A simple sample preparation procedure was developed to improve the conventional method in metric-assisted laser desorption ionization mass spectrometry by reforming the sample morphology during the drying process. The advantage of this technique is that the signal intensity of carbohydrate samples prepared by this optimized method can be effectively enhanced.
Generally, individuals new to this method will struggle because the methanol deposition step is very crucial, and it needs to be done in one go. Any hesitation will decrease the data quality.
To begin, while wearing nitrile gloves, use 100 milliliters of detergent solution to hand wash the sample plate. Use distilled deionized or DD water to hand wash the plate. Then, with 30 milliliters of methanol, rinse its surface. Insert the sample plate into a 600 milliliter beaker and fill it with DD water until the plate is fully immersed. Then place the beaker into an ultrasonic bath and sonicate the plate for 15 minutes. Remove the sample plate from the beaker and use pressurized nitrogen to blow off the water drops. Then deposit 0.2 microliters of methanol on the sample plate to check whether it spreads to other spots. If it does, repeat the wash, sonication, and nitrogen treatment as just demonstrated. After preparing the drying chamber according to the text protocol, premix 0.25 microliters of 2,5-dihydroxybenzoic acid solution and 0.25 microliters of sialyl Lewis A or maltoheptose solution